top is unsaturated. What do you look for to know that? The double bonds. Remember, on the test, to save space. To save space. They may not draw it all kinked up like that. The double bonds cause kinks, which makes it a liquid at room temperature. However, it might not actually be drawn all bent. It could be drawn in a straight line. Look for the double bonds. In fact, the more double bonds, the more likely it would be a liquid. It would actually, one double bond would make it, let's say, a liquid at room temperature. Two double bonds might make it a liquid even at a little colder than room temperature, if that makes sense. More double bonds, the more likely a liquid even at low temperatures. Yes?
How can you tell if it's a trans fat?
You won't be asked about that. Technically, a trans fat would be like it would go up and then come back down, and it would sort of form a straight line rather than bending like that, but there won't be any questions about trans fats. I just threw that trans fat thing in there because they talk about it so much.
How do you know that it's a fatty acid?
Oh, okay, so fatty acid, it's a long chain of carbons with a carboxyl group, which is also called a carboxylic acid group, and that's why it's called a fatty acid. When 3 of them connect to a glycerol, that's when we get what? A triglyceride. Now, here's another question. Which side of this, the left or the right, would be the side that would attach to a glycerol? The right side. In a lot of the pictures, in fact, and I didn't do this on purpose, but in every other picture I've shown you, it's attached at the left. That's because this was always on the left. Here's a diagram showing a dehydration synthesis, and usually when they're showing this, they draw it this way. But notice what it's reacting with. It's reacting with that carboxyl group. What's going to be lost? Water, right? In this case, 3 waters, and then that's where we're going to get our triglyceride. Okay? So it would attach on the carboxyl side. In that drawing on the previous page, obviously that was on the right. I didn't do it on purpose, but I just wanted to clarify. It's not. This would be happening in 3 dimensions. It's not like it would orient itself so that it was on the left side of the room in order to react. It would be happening in 3-dimensional space. All right, what about this? What's this? It's that is a phosphate. It's not a phospholipid though.
Steroid.
It's not a steroid. Look, this formula is C5H10O5. So this would have to be a sugar. Happens to be a 5-carbon sugar. It could be ribose or deoxyribose. And what is this? What's the 3rd part of a nucleotide?
Oh, I see what it is now.
Do you really?
It's a hexagon.
A hexagon with nitrogen in it. Right, so this is called a nitrogen base.
Oh, this is the chunks.
This is and I did talk about chomps. Somebody just said that.
So chomps, right?
Part of chomps. I told you that carbon, hydrogen, and oxygen, the CHO part we see in carbs and lipids, and then I'll answer your question. In proteins, we see CHO, and S, the sulfur for those, we'll talk about that. And then in DNA, we see CHON,
P.
and then the P, the phosphate group. And not just DNA, but in nucleic acids in general. Question?
I want to know all this breakdown.
You should know. If I showed you that picture, I could ask you, what is this? What is this? And you have to tell me it was the sugar, the base, the phosphate.
How would we know it's sugar? Yeah.
Because it would only be, I would never, let me put it this way. This test is, if you're supposed to recognize that that's a nucleic acid, there's always the same 3 parts of a nucleic acid. It's always the phosphate connected to the sugar and then the nitrogen base. If you look at that biochemistry study guide handout with all the pictures, it has this exact thing. It has all of them. As far as sugars, this is a chain of sugars, which is called a polysaccharide. What do you say, saccharide?
No, he only says monosaccharide.
Oh. So this is a polysaccharide. It's the only thing that looks like a chain, but that's not connected. Remember, a steroid looked more like this. It was 4 rings, and they were literally fused, something like this. They were literally fused to each other. This is a polysaccharide. These are monosaccharides. They don't draw in the carbons, and we've talked about this too, how these are actually all carbons here. They just, for shortcut purposes, same thing here. They just don't draw in all the carbons. They just draw it if it's something other than a carbon, like an oxygen or a nitrogen or something like that. All right, so how many sugars are in this polysaccharide? 4. Okay, yeah, that was the easy question. Now comes the harder question. If this is a 6-carbon sugar, What would be the formula for this polysaccharide?
C6H12.
No.
Not for the whole thing. C24,
H48.
H48,
Oh, okay. I thought you were starting from.
O24. But actually, that's wrong.
Okay.
Here's why. I told you that for monosaccharides, it is perfect 1 to 2 to 1. But what happened to connect these? We lost an H2O. And we also lost an H2O here. And we also lost an H2O here, right? 3 connections. So technically, what is actually the formula for this? We didn't lose any C's.
42.
H42,
21.
O21. Because it took 3 waters. And what's that called? The water release?
Dehydration synthesis.
Condensation or dehydration synthesis.
Where's the C8, like C6? What's the
Any monosaccharide will be C5H10O5, C6H12O6. So if it's just one,
2 to 1? Well, 1 to
it'll be the 1 to 2 to 1 and it's a monosaccharide. For a poly, it will be off by a little bit.
1 to 2 to 1. 6 to 1.
Right.
6 to 1 to 1.
Alright. This one. I know this is a little hard to see. And on the recording it's going to be all stretchy. But can you tell me what this is?
Amino acids.
This is a chain of amino acids, yes. How would you count it? You had to know how many. How would you figure it out?
NCC.
Look for the NCCs. So NCC, NCC. I think this one is actually 8. I just found it online. So here's 1, 2, 3, 4, 5, 6, 7, 8. What the heck are all these things sticking off? R groups. It's just that they actually drew in the R group. So don't let that throw you off. You see all this crap hanging off of it. They just replaced R with what R really is, which is one of 20 different things.
So to find how many amino acids.
You literally just go NCC, NCC, NCC.
And that makes up the protein.
Yeah, and this would be part of a protein. It probably wouldn't be a whole one. And the real ones are really long. But what level of structure would this be?
Primary.
This is showing primary structure. It's just the order of amino acids. We're not showing how they bend or fold or anything. Now, what's responsible for secondary structure? Hydrogen bonds between what?
Carboxyl group.
And
Hey.
every amino acid, and you have to know this, has a carboxyl group on one side, that's a C, and an amino group on the other side. The secondary structure is formed by reactions between the hydrogens on these and the oxygens over here. In other words, let's just say this hydrogen makes hydrogen bonds to this oxygen. And let's say now this nitrogen does the same thing. And now this nitrogen does the same thing. What are you going to get? You're going to get some kind of a repeating pattern. That's the secondary structure. It's reactions there. Now what's going to happen after that is reactions between what? What's tertiary?
R groups.
The R groups are going to start reacting with each other, and that's when you took the pipe cleaner when we did the exercise and you kind of crumpled it. Because now what's going to happen is maybe this R group is hydrophilic and it forms a reaction with this one, and maybe some other one's hydrophobic, so it they orient themselves. And especially this one I told you about, the sulfhydryl group. They form bridges across. What would you be looking for in a quaternary structure?
More than one.
Right, multiple chains. There have to be more than one chain. All right. So yes.